And so to introduce this, what I'll do is just provide you with a sort of real-life story. And this is something that came out of the news about 6 months ago, where there was a young child who suffered from actually a brain disease called Joubert syndrome, where basically she was prone to falling asleep, and then her heart would stop while she was sleeping. Nothing was wrong with the heart, but she had this neurological disorder that she would eventually die from. At the same time, there was another child who had a congenital heart disease that needed a transplant. And so the idea was that because this child would not survive, that they would take her off the respirator, and that she could then serve as a donor then to the other child who had this congenital heart disease. The complicating factor here was that when they took this child off the ventilator, that she actually did not die right away, and then this created a series of ethical problems in terms of eventually transferring the heart to this child. Eventually, there was a donor found for this child, but this sort of underscores the problem here, that when you want to do a heart transplant in a young infant, it's very difficult because there simply aren't a large number of donor hearts that are available. And then you have to also worry, of course, about blood group compatibility. And so what we'd like to do is develop technology then that could get around the blood group compatibility problem, and perhaps also extend the window within which you could carry out a transplant. And so most people, I think, are aware of the fact that if you're going to do an organ transplant, it's very important to match blood types. These are called the ABO blood groups that are important. You can actually do things like kidney transplants and liver transplants, where we don't have to be so careful about this. However, in heart transplants, which is what we're interested in, basically every attempt that's been done in an adult has been fatal, and that it leads to what's called hyperacute rejection, essentially where your immune system attacks the organ that's been implanted, and then you eventually die from the immune response. Several years ago now, our collaborator Lowary West showed that, however, that if you take an infant or a newborn, a child who's in their first 6 months of age, that you actually can do heart transplants across the blood group barrier. And it was thought that basically when you implant this heart, that before the immune system is fully developed, that the child develops in the presence of that foreign heart and does not recognize it as foreign, and so therefore there's not an immune response that develops. And so the idea was that could we somehow expose a newborn to these blood group antigens attached to either nanoparticle or a stent, and that this would induce the same, what we call tolerance, and this would then allow eventually transplants across the blood group barrier. And so the idea was that we would prepare a device of this sort, which we would then have the blood group antigens on. Sometime you would identify a child who had a need for a heart transplant at some point later in their life, or perhaps even sooner, very soon. And so you'd implant this device, and the critical thing here would have to be done early in the child's life before its immune system had fully developed. This would then, the immune system would develop in the presence of these foreign antigens. This would then extend the window with when you could do a blood type incompatible organ transplant. And then at some point later, when there was a donor heart that was available, you would be able to do the heart transplant. The idea here was that by making this implant, you would extend this window in which you could wait until a donor would develop, and so that would potentially increase the donor pool. All right, and so what would the market here be? Well, this would be basically infants who are in need of a heart transplant, and here's some numbers for you. There's about 1 million babies that are born worldwide each year with some sort of congenital heart defect. About 10% of those will die every year. And about 1 in 100 infants have a congenital heart disease to some extent. So if you look more locally in terms of North America, these are US numbers. About 1 million, between 650 and 1.3 million Americans have a congenital heart defect. And there's a very substantial number of individuals, around 40,000 babies are born each year with one of these defects. And many of them will die before their first birthday. So what we'd like to do is develop some methods to help allow these transplants to happen later in life. So these are the blood group antigens. I won't talk too much about the chemistry, but basically if you're blood type O, your blood cells, as well as virtually every cell in your body, has a carbohydrate structure like this. If you're blood type A, then this structure is further elongated by the addition of this red sugar. If you're blood type B, then you have a similar structure to this, but now you have the blue sugar here, and you'll note the only difference between the two is simply you have this, what we call an acetoacetal group here versus a hydroxyl group. So it's a very small difference between these two. But that's the whole basis for this, the specificity of the immune recognition of the blood group antigens. And so the idea was that what we wanted to do was synthesize these antigens. We then wanted to attach them to some sort of stent or nanoparticle. And for this, we're using silicon-coated materials, either silicon-coated stents or silicon nanoparticles. Join these together, and then we would have an antigen-coated material. And so We have looked at stents, and this has been successful, so we'll take these surgical stents and then we'll do coating with silicon, and then are able to attach antigens to that. What I'll focus more on today is really the nanotechnology aspects of this, where we've actually made nanoparticles. And so some of the advantages of nanoparticles in particular is that they're more easily tailored than the stents. For example, we can influence their size, their loading, or also the material from which they're made. They can be labeled for certain properties. For example, you could imagine in certain imaging applications it might be useful to have either fluorescent or magnetic nanoparticles. So here's an example of, or show you here a series of different nanoparticles that have a fluorescent dye under UV light, so you can see that there's a fairly bright fluorescence here. The other big advantage of a nanoparticle over a stent is that you wouldn't have to surgically implant this. A stent, of course, would require surgery to be put in, and so these could then be injected into the bloodstream. The other advantage here is this would allow a regimen in which you could actually provide multiple treatments over a series of say, weeks or months even, again using IV injection. So we think both technologies have promise. I think we're more at the moment more excited about the potential of the nanoparticles. And so what we're using here is silicon nanoparticles. And so basically we take these triethoxysilane, generate then these silicon nanoparticles, which I'll show as this structure here. We can tune the size of these. These are shown, this is an SEM image, where here what you see, these are about 100 nanometers in size. We can make these larger or smaller if we wish. And again, that's one of the advantages of the nanoparticles. We then functionalize these nanoparticles further using then this amine containing material. So what we can do is then coat this with a series of amine groups. These are reactive functional groups. We can then use this basically as a handle for attaching our antigens, which are these synthetic carbohydrates. So here what we do is we take our nanoparticle that has an amine on it, add some sort of antigen. In this case, it's a carbohydrate blood group antigen through some sort of activated ester. And this then generates our silicon nanoparticle that then has carbohydrates on it. And this same approach could be used basically to label any sort of any nanoparticle with any antigen. And so that's where we sit at the moment. And so basically we've developed now some fairly robust methods for coating nanoparticles with virtually any antigen. We are trying to do, however, some optimization of this chemistry, and that's currently in progress. We're now hoping to move into some animal studies. The first thing we'll do will be injections in mice to see whether we can get tolerance in mice. And then finally, the next thing we'll do is work in pigs. As you could probably imagine, it's not so easy to get a pig experiment started in the current climate of H1N1. And so we had actually planned to move into pigs directly, but mice is where we'll be starting. It turns out that pigs are actually a better model for humans. But nevertheless, the mice studies are actually going to be started actually very soon, so right after the new year. So in terms of where we sit with the timeline of the technology development here, we have filed a provisional PCT application on this technology. That was last year. We're now in the process of filing a full patent on that. We've sort of developed the initial prototypes. We'll continue the animal studies here, and then hope then by in about 4 or 5 years now, well, 4 years I guess, have some sort of product out that we could start to develop. So with that, I'll stop and just again, I should point out that if you're interested in this technology, the person that should be contacted would be Dr. Elizabeth Nanak, who's the Associate Director for Business at the ASCCS, and this is her email address. So again, thanks very much for the invitation.